So we're moving on to our final lecture of the morning. Jack O'Meara is coming to talk to us. Jack is from Athlone, so he graduated as a biomedical engineer in Galway, and has joined up with a doctor to look at the liver and therapeutics for the liver in really innovative fashion. And so I look forward to hearing. Now, he's promised he'll try and dumb this down for us, because I think it's a bit complicated.
It's not very complicated. It's not very complicated. And sort of the other side of the spectrum, we're at the very early days of trying to make medicines for diseases that have not yet been so inspiring, had such inspirational cures. And we're an early stage drug discovery company focused on the liver, so broadly focused on the liver across indications where we think we can have use new tools like RNAi and genomics and human tissue models to try and improve the efficacy of medicines in our space. Unfortunately, we don't have any stories about amazingly inspiring 84-year-old women who are fighting the good fight to cure patients, but I'll try and give you a bit of science, and hopefully that will keep folks engaged. So I need to turn on this guy. Okay, so RNAi, liver medicine, sort of discovery stage. That's sort of the name of the game here at Ochre Bio. Done. Okay, so I think most people in this room appreciate that liver disease has been, particularly chronic liver disease, has been majorly on the rise for quite a while. This is also my investor deck, so I try to really illustrate that very clearly, that even though we've seen a lot of progress in things like cardiovascular disease and cancer, we really haven't seen a lot of treatments, bar in the sort of Hep C and Hep B space. We haven't seen a lot for more chronic versions of liver disease. And we think that there's been a lot of trials. There's been a lot of, particularly for things like fatty liver disease, NASH, there's been a lot of efforts made, but nothing has really worked in the clinic. It's seen as almost a graveyard for drug developers, a bit akin to Alzheimer's almost in some ways. But we think, when we started the company about 4 years ago, that there's a convergence of new technologies that are really allowing us to improve the probability of success for discovery and for development of medicines in this space. The three things I think are really impactful, and which we've built our thesis around, are that new tools like advanced genomics, single cell, spatial sequencing, they give us a much higher resolution into the biology and to try and understand what's going wrong, and computational techniques to mine all that data are increasing our powers. Also, a drug modality called RNAi. It's a bit like mRNA, but it's sort of the inverse. Rather than turning on a gene, you turn off a gene. And the beauty of RNAi is that it's highly specific. So mRNA goes everywhere. That's great for vaccines, not so great for very targeted therapies, whereas RNAi is very highly specific to the liver, which particularly attaches sugar to it, which we do, which is great because it can be very targeted. And then lastly, advances in clinical models. So animal models, every drug that was tested in a patient for liver disease ultimately failed, and particularly for NASH and otherwise. But a lot of those worked in mice, so the mice aren't really telling us the right answer. We got to think differently about how do we do translation, how do we use more human-centric models to really give us the confidence that our medicines will work. So we built a drug discovery platform that spans a lot of in silico drug design. So we do a lot of work around generating proprietary data on liver disease progression using these advanced genomic tools, and then have various computational folks internally, or the sort of way smarter than me, AI whiz kids, to look at all that data and then try and map it to phenotypic analysis. So things like clinical variables, things like histology samples, et cetera, to try and tease apart a picture of what's going wrong. Then we can very quickly design these siRNA medicines. It is a bit, I don't know if you've heard much about the mRNA, but they are almost programmable. Once you know the gene or the sequence of the target that you're interested in, within a couple of weeks, you can spin up a potential drug candidate, and then you can go directly to testing it in a relevant translational model. And that's sort of different than traditional drug discovery. Traditionally, if you can have a hypothesis about a gene that could be relevant to a disease, and then small molecule drug discovery campaign could take a couple of years before you can ever even test it in a relevant preclinical model, a mouse or otherwise. Whereas we can go from hypothesis, biological idea, drug, testing it directly in human liver samples in a matter of weeks, which we think will be a real, so it allows us to speed up the iteration cycle as we move through different sort of hypotheses and what drugs will be most effective for patients. And then lastly, we do a lot of work on the models side, models of disease. So we do a lot of work in Oxford in cell models, so taking primary human hepatocytes, kind of making 2D cell models, co-culture models, varying ways to try and study the biology and study whether or not we think our medicines have an effect. But of course, they are models, particularly cell models. You kind of just clump of cells together and hope that's telling you something about the disease. So we go another step further and we take biopsy samples of diseased liver tissue and we slice them up into slices, and then we can test whether our medicines are working in a slice of diseased liver tissue. And the beauty there is that you're not sort of inducing injury or inducing disease. It's actually a diseased tissue sample, so we hope would have sort of stronger ability to recapitulate the human disease state. But of course, it's a slice and it's been taken out of a body and it's not that happy by nature, so it's only so good. And then the sort of crown jewel of our whole kind of approach to validating these medicines is we actually work with hospital systems, particularly in the US, to retrieve discarded donor organs. So organs that would have been offered up for transplant, but for various reasons were non-usable, typically due to steatosis or typically due to having sort of too much fat, which is the kind of disease we hope to one day be able to treat. So we take them, we have what we call our organ ICU. So it's effectively a surgical staff on hand to retrieve marginal donor livers, put them on these new perfusion devices, which are like a big sort of pump that tries to, I heard someone once call it a Frankenstein machine. It sort of pumps blood and oxygen and through the liver and has a kidney dialysis unit attached. And it's very sort of trying to keep the liver as happy and believing it's still in the human body as possible. We do that for about a week and we can dose the actual diseased human liver with our intervention and study any physiological changes or any response to the treatment. So it's a very novel way to think about making new medicines. And the thesis is really we're almost doing a clinical trial before the clinical trial to make sure this medicine will have the effect that we want it to have. And then from there, move into human testing. We do animal studies for long-term safety. We do work in mice and potentially may do so in larger animals, but we're trying to minimize the amount of work that we do in animals, not just because animal testing isn't a great thing to do for society, but largely because it's not a great model of human disease. It doesn't really tell us the right answer. So we think having a more human-centric approach will lead to better medicine. So that's the thesis. I won't spend too much time on this, but just to give you a sense for sort of the data science side of our company, because it's a big sort of founding thesis, is when we started in 2019, we did a very large spatial sequencing atlas of 1,000 liver samples across the disease continuum, where we basically used a histology AI company to take a picture of the liver sample and then zoom in on different parts of it and then sequence the transcriptome with those particular sections. It basically asks the question, in a more diseased section of the same tissue sample, what genes are upregulated or downregulated? And it could be driving that disease progression. And then we can map that not just to the histology, but also we do about 150 clinical variables on those patient samples. So you kind of begin to build this sort of robust picture of the disease process and come up with hypotheses around which genes may be playing a key role and could ultimately be good drug targets to develop medicines against. We do a lot of single cell sequencing. Our CSO sort of was big in the single cell world when I was getting started in the UK. And that gives us a really deep view of which cell populations are potentially contributing to the disease. And we know that our drug modality is it's a GalNAc siRNA, so it's very goes directly to hepatocytes at a very high specificity. So we know we can then prioritize genes that are more highly expressed in hepatocytes, et cetera. And then the latest and greatest, and this is definitely our CSO's side of the business, but we do a lot of phenotype-free screening. So basically we use CRISPR in primary human hepatocyte cell lines to knock out a particular gene and then study how that changes the overall transcriptome. So like the overall what's being read. And then we can try to look at what pathways have changed when we knock out a particular gene against all of our existing data set and then see if there's some phenotype or see if some sort of pathway association between, oh, that gene is sort of a non-obvious relationship with that pathway. Maybe we should go investigate that further. And that's when all the AI gurus come into the company, start talking very scientifically about how we use these technologies to try and improve our drug discovery hit rates. So I mentioned a bit, we use it a bit in sort of the histology side of the house, but again, this is an investor presentation, so everyone wants to hear about AI right now. It's a hot topic. But in its application in target screening, an example case study we used recently, which is on the bottom right-hand corner of the screen, is where when we would nominate genes for our primary human hepatocyte model screening and see if we're sort of preventing cell death in a 2D cell culture model, we typically get about 1 to 5% hit rate that those genes would have the desired effect on the model itself. But when we use AI applied to these large data sets, we can actually increase and then help that recommend targets for screening, we've actually been able to increase that by 25 to 40%. So the kind of computer is seeing what the phenotype we want to get out is, and then saying which of the pathways that we've studied seem to have the highest relationship with that phenotype. In this context, it was sort of cell death, and then really helping us make better decisions early on in our drug screening discovery stages. And the hypothesis there is that because we're not doing big, very large scale high throughput screens or even very large scale mouse studies, or otherwise we're using these sort of inherently low throughput models like discarded donor organs, which there's a very finite amount of, and we want to have really high confidence that the genes or the drugs that we're moving into that phase of our discovery process are going to have a high probability of success. So we use these tools to try and make us smarter sooner, and so forth. So just on I mentioned this a bit already, but just on the earlier side, the two models that we use, one is sort of various different cell culture models for the initial screening, initial testing of a particular gene or a particular siRNA. I use those terms slightly interchangeably, which is sort of a bit silly, but basically once you know the gene, once you believe strongly in the gene, it's very quickly to develop an siRNA to knock down the particular sequence of that gene. So they're kind of almost the same concept and used kind of interchangeably in our company. And then we do a lot of work in Taiwan in precision slice culture, where we can look at different phenotypes that we can induce or study just directly in slices of a diseased liver biopsy. But then, of course, the crown jewel, as I mentioned, is this whole organ perfusion group that works out of the US, where we take about 100 livers a year we've been able to do, where we can study at a very high fidelity whether or not our medicines are improving and we look at about 50 different markers. We're throwing the kitchen sink at this model because it's very low throughput, but we hope we'll have good insights into the biology. And this is just some of the data we then begin to look for. As we've been doing this for a few years now, and it's very complex work because it is a human organ on a machine. It's sort of inherently a bit unhappy, and there's a lot of variability in the different sort of signal that you get out of the model. But we've looked at different parameters that seem to consistently or stay relatively consistent within the context of the 5 days that we tend to keep these livers going on the machine. So this is sort of various different markers that we've looked, and the gray dots represent the sort of baseline. We're talking about 70 livers that just as a baseline data set to be able to then try and study whether or not our intervention are changing that. And each day there's sort of various different samples taken. And then we've introduced a target X, which is one of our lead candidates that we've been working on for the last couple of years. We have about 6 that we're very excited about and seem to look quite well across the various models I've described. And this one we've seen has an improvement on the necrosis cell death when the liver is on the machine over a few days. So we've been able to see that by treating the liver. It's very small studies, we're just beginning, but at least there's some signal that we can reduce necrosis on the machine over the first 5 days. We've also found that portal flow of the liver while on the device is sort of a very sensitive marker to intervention. And we've seen that we've been able to improve portal flow by introducing it. This is a novel gene that we've identified using our technology and designed an siRNA for, and seem to, at least in this initial study, improve the portal flow. And then similarly, we don't see any major sort of concerns with lactate clearance or BUN, so no major reduced liver function. So this is sort of early data that would give us confidence that maybe this novel biological target could actually play out to be a meaningful medicine, and one day have the type of effect that some of the HCC studies have shown. So then we used the various models to try and build a bigger picture of if we consistently see the same effect for this particular drug candidate. And this is actually done preceding to the perfusion, but I think the slides are mixed up, my mistake. But anyway, we've seen that knocking down a particular gene across multiple different donors or multiple different patients consistently prevents against cell death, which is one of the indications we're looking at is a ischemia reperfusion injury in a liver transplant context. We're preventing against that cell death. And then similarly, we see that it's very consistently knocks down the gene of interest across multiple different donor types in the precision slice model. So we know the drug is doing what we think it will do, and we're going to have a stronger confidence that it's not going to be a drug effect. It will be whether or not the biology plays out when we test it in the human organ. That's really it. I don't want to spend too much time, but hopefully give you a bit of a taste of what we're trying to do. We're still working through our clinical strategy. I think one of the things that's a challenge for companies like this who've got a very robust scientific platform is trying to figure out exactly where to first enter the clinic. I think particularly our journeys with these types of companies. You got to raise a bunch of money to build the kind of infrastructure to do this type of discovery, and then everyone's waiting for you to say, okay, show us it works in a clinical trial, and then your first clinical trial becomes really, really important to make sure that you have a high probability of success, so that you can continue to invest in the product and continue to invest in the sort of scientific founding hypothesis. So we're very focused on indications where there's sort of a clear need and a relatively well-defined sort of endpoint that we think we'll be able to modulate effectively with the drugs that we're working on internally. So we're looking at a few various decompensation cohorts or subsets of patients who are experiencing decompensation to prevent further decompensation. We're also looking at an indication in liver transplant, because there is a lot of livers that never get used, despite potentially having, and despite the major unmet need on the waiting list for liver transplant. So maybe we'll be able to use this technology to try and rescue marginal donor livers for transplantation. But ultimately, the vision for the business is to move upstream into more chronic conditions, things like NASH. But the problem with a clinical trial in NASH is that it's very, very expensive and very, very long term. So until we can demonstrate effect of these medicines in sort of shorter term initial cohorts, we won't be able to raise the money to go after the larger diseases. But that is definitely the sort of longer term vision for the business. And then ultimately, I think a very nice vision of how medicine changes in the liver disease space, obviously this is a long way in the future, but is a bit like how statins have really rechanged heart disease by almost being preventative interventions prophylactically prescribed to patients. And we know that liver disease, really chronic liver disease, is inherently tied to your age and sort of your weight. We know some of the preemptive risk factors. So if we could develop interventions, really highly specific, highly safety profile interventions like these siRNAs, maybe we could keep metabolic function healthier for longer and ultimately sort of get ahead of the degenerative symptoms of liver disease. But anyway, that early days in our business and trying to make the kind of impact that we just heard before me. So looking forward to taking any questions. Oh, last, I have one more plug. Sorry, I forgot. My last thing was I live in the UK, but I grew up in Athlone, as was heard. And I think Ireland is this amazing sort of home to particularly large biopharma and larger biotech companies, but not many of them are indigenous biotech companies. But there is a few early stage biotechs that are blossoming in the country. And I think Ireland has amazing opportunity for all the wonderful research and talent in the room, a lot of the industry experience of the companies that have came here in the 80s, 70s, 80s, 90s, to try and merge those two worlds and spin out indigenous companies based off of the research that's emanating from universities like the one we're in today. So anyway, my plug for, if you're out, start a company, get out there, trying to make some medicine, sort of make the world a better place. I think it would be great to see the Irish indigenous biotech sector grow as well. Anyway, that was my last little plug.
That was a wind whirl.
Sorry, we're going a bit fast, yeah.
No, but it was brilliant. It was really I was expecting to be like trying really hard to keep up and not understand. And actually you made it very clear for those of us who are not in the area. So thank you very much. Any questions from the floor?
Yeah. Hi.
Hi.
Your crown jewel ex vivo model is
Yeah.
very exciting. Are you also looking at dynamics, pharmacokinetics in that, or just efficacy? Or
So we are, I had
Yeah.
a slide, I think I took it out, just worried about time, which I then sped through the whole presentation. We do look at pharmacokinetics. That's one of the key sort of parameters that we can study very effectively. The challenge with a model like that is you've only got 5 days, right? So trying to see efficacy signals, you're only seeing sort of trends towards efficacy, but you can very accurately measure where the drug is going and is it getting into the right cell type? Is it sort of beginning to do what you think it will do? So yeah, pharmacokinetics and
are you looking at different routes of administration, or?
so these
introduced.
siRNAs are actually subcutaneous. So they're the other kind of more well-known drug that uses the same approach is a drug. I don't know if it's been launched in Ireland. It was launched in the UK recently called Leqvio for cholesterol. It's basically a once every 6-month jab in the arm, and it goes very specifically to the liver and modulates liver proteins in a very highly specific way, which is great. So we use the exact same modality, but we're just trying to use it for more liver disease indications and looking at new biology or new targets. No worries. Thanks so much.
Thank you. I don't know much about livers, and I could follow most of that. So you did a good job.
Despite the speed.
I'm just interested, if you work in labs and just do slices of livers,
Yeah,
how do you know about any ongoing systemic side effects?
that's the key. Even with the whole organ perfusion model, you've only got 5 days, and it's an isolated organ on a device. So we're trying to see trends in the efficacy of the medicines that we're trying to see them do basically what we think they'll do in the right way. But we do long-term animal testing, primarily for safety, to make sure there's no off-target effects. It's not having any sort of weird, there's nothing negative happening as a result of introducing this drug. But the problem with animal testing is it hasn't given us a good view on whether or not the drug works. It'll tell you pretty well that it doesn't do anything negatively, but that's why we're focused on, when we pick the medicines we think will be most efficacious, we're using the human data, but to be sure there's no downstream side effects, we use animal models as well, but in a more reduced fashion.
Yeah,
That makes
that's if it had occurred to me, it had probably occurred to you as well.
Yeah.
But I also want to ask, when you do a clinical trial, where do you go for your money? Who funds?
Yeah, so it's
your big
mostly
trials?
So to date, we've raised almost all our money in the US. We raised a bit of it in the UK and a bit of it in Asia, but a lot of the sort of high risk, this is a high risk type of business. It's like, it's very expensive, and it's obviously a big end game if it works, but it's very high risk inherently. So there's a lot of risk. The risk appetite in Ireland, but Europe more generally, tends to be a lot lower. So we've had much more success by pitching this story to US investors who are a bit more big picture focused. Yeah, no worries. Thanks. Yeah. My CSO, yes.
Dr.
Wills, yeah.
So has that combination of, obviously he comes from a medical background.
He's a medical data science guy. It's a weird combination. He worked as a doctor in South Africa, originally focused on alcoholic hepatitis. This is what are we calling it, pre-millennium? Last century. And I heard that joke used a couple of times. And I came from initially biomedical engineering research, and then I worked in the US for a few years in sort of early stage biotech companies, helping to launch products for them. And my role is more on the sort of fundraising, company building side of the house. He's more the data science, early discovery work, and we kind of meet in the middle for the clinical.
On your website, there's a global piece talking about the global liver aspect, and it's sort of mentioning is there global, I guess you talked a little bit about how we reach other populations, there's liver, in the less economically regions. Is there any kind of, I know it's way early in your disease, you've not even got your medication that's utilized.
We haven't even picked a drug yet. No, but I think one of the things about, I worked in rare disease before, and rare disease is an amazingly important area with a huge unmet need. And fortunately, over the last 10 to 20 years, there's been major breakthroughs, but it's completely inaccessible for most of the developing world because the price of these medicines is so high. So because we focus on siRNA, it is a very low cost product to manufacture. Like the cost of these gene therapies are astronomical just to make them, let alone sell them. Whereas using sort of a programmable medicine, the same way that mRNA is a very low cost medicine to make, the vision is to be able to make a product that is widely applicable across the world.
Thank you very much.